Repair and Regeneration. Introduction. Healing is the body response to injury in an attempt to restore normal structure and function. Healing involves two distinct processes. First, regeneration, second, repair. First, healing by regeneration. Healing takes place by proliferation of parenchymal cells and usually results in complete restoration of the original tissues is called regeneration. Regenerative capacity of different cell types. The cells of the body are divided into three groups on the basis of their regenerative capacity as following. First, labile cells. Second, stable cells. Third, permanent cells. First, labile cells. These cells continue to multiply throughout life under normal physiological conditions. Example, surface epithelial cells of epidermis, cells of lymph nodes and spleen, etc. Second, stable cells. These cells decrease or lose their ability to proliferate after adolescence but retain the capacity to multiply in response to stimuli throughout adult life. Example, parenchymal cells of organs like liver, pancreas, etc. Third, permanent cells. These cells lose their ability to proliferate around the time of birth. Example, neurons of nervous system, skeletal muscles and cardiac muscle cells. Control of cell growth is by growth factors. Regeneration of any type of parenchymal cells involves the following two processes. First, proliferation of original cells from the margin of injury with migration so as to cover the gap. Second, proliferation of migrated cells with subsequent differentiation and maturation so as to reconstitute the virginal tissue. Second, healing by repair. Healing takes place by proliferation of connective tissue elements resulting in fibrosis and scarring. At times, both regeneration and repair takes place simultaneously. Two processes are involved in repair. First, granulation tissue formation and second, contraction of wounds. Repair response takes place by participation of mesenchymal cells, endothelial cells, macrophages, platelets, and parenchymal cells of the injured organ. The conditions in which tissue healing is achieved by repair. First, when recovery fails to occur in an acute inflammation. Second, when parenchymal cells necrosis cannot be repaired by regeneration. First process involved in repair is granulation tissue. It is a highly vascularized connective tissue composed of newly formed capillaries, proliferating fibroblasts, and residual inflammatory cells. The term granulation tissue derives its name from slightly granular and pink appearance of the tissue. Granulation tissue typically grows from the base of a wound and fills the injured area while the necrotic debris is being removed. Roles of granulation tissue. First, anti-infection and protecting the wound surface from further injury. Second, filling incision or wound and any defect of tissue. Third, replacing necrosis tissue, effusion and other foreign body. The following three phases are observed in the formation of granulation tissue. First, phase of inflammation. Second, phase of clearance. Third, phase of ingrowth of granulation tissue. First, phase of inflammation. Following trauma, blood clots at the site of injury. There is acute inflammatory response with exudation of plasma, neutrophils, and some monocytes within 24 hours. Second, phase of clearance. Combination of proteolytic enzymes liberated from neutrophils, autolytic enzymes from dead tissue cells, and phagocytic activity of macrophages clear off the necrotic tissue, debris, and red blood cells. Third, phase of ingrowth of granulation tissue. This phase consists of two main processes. First, angiogenesis or neovascularization. Second, fibrogenesis. First, angiogenesis. Formation of new blood vessels at the site of injury takes place by proliferation of endothelial cells from the margins of severed blood vessels. Initially, the proliferated endothelial cells are solid buds, but within a few hours develop a lumen and start carrying blood. The newly formed blood vessels are more leaky, accounting for the edematous appearance of new granulation tissue. Soon those blood vessels differentiate into muscular arterioles, thin-walled venules, and true capillaries. Angiogenesis takes place under the influence of following factors: vascular endothelial growth factor, platelet-derived growth factor, basic fibroblast growth factor, and other cytokines are all associated with cellular proliferation. Second, fibrogenesis. The newly formed blood vessels are present in amorphous ground substance. The new fibroblast originates from fibrocytes as well as by mitotic division of fibroblasts. Collagen fibrils begins to appear by about 6th day. As maturation proceeds, more and more of collagen is formed while the number of active fibroblasts and new blood vessels decreases. This results in the formation of inactive looking scar known as cicatrization. Second process involved in repair is contraction of wound. The wound starts contracting after 2 to 3 days and the process is completed by 14th day. During this period, the wound is reduced to approximately 80% of the original size. Contracted wound results in rapid healing. Factors involved in wound contraction. First, dehydration of wound. Second, contraction of collagen in wound. Third, appearance of myofibroblast.